Hi. In multiple sequence alignment, we are comparing three or more sequences at the same time. It's actually an extension of pairwise alignment. In this video, I am going to talk about the principle and algorithms used in multiple sequence alignment. Multiple sequence alignment is an extension of pairwise alignment. It is used to align multiple related sequences to achieve optimal matching of the sequences. So in pairwise, we are considering only two sequences at the same time, but in multiple sequence alignment, we are considering three or more sequences. Maybe we will be considering 5 sequences, 10 sequences, or in the database searches, we are considering the pairing between thousands of sequences at the same time. So the algorithm requirements or the software requirements for doing the multiple sequence alignment is way much more than the pairwise alignment. It actually what it do is it is converting multiple pairwise alignment into a single alignment. So the basically what they are doing is the pairwise alignment, but these pairwise alignments they do a stacking and make into a single alignment. So this multiple sequence alignment actually reveals more evolutionary relationship between organisms than a pairwise alignment. So this is an example of the multiple sequence alignment. They are fitted into the same frame to maximize the similarities between different sequences. There are different advantages for multiple sequence alignment when we are comparing it with the pairwise alignment. It reveals more biological information related to evolution than the pairwise alignment. It identifies the conserved sequences, which is especially important for the promoter region, the functioning of gene on the protein structure. It identifies critical amino acids, which is essential for the catalysis or binding. It's used in phylogenetic analysis to study the relatedness between protein sequences and nucleoside sequences. It helps to predict secondary structure, tertiary structure, as well as the function of the protein. And it is used for primer design. Multiple sequence alignment is to arrange sequences in such a way that the maximum number of residues from each sequences are matched up. So we are looking for maximum similarity. In sequence alignment itself, we are looking for similarity. And if you are doing a pairwise alignment, we want to have the two sequences have maximum similarity. When multiple sequence alignment, we want to have all the sequences we are comparing should have maximized similarity. We can add gaps, or we can add gap penalties and everything to make sure that the optimal solution has been obtained. And there is a scoring system for multiple sequence alignment. It's called SP scoring. It is sum of scores of all possible matches, and there will be a consideration of the gap penalties if when we are adding, just like in pairwise alignment, when we are adding one gap, there will be back penalties, and the sum of this matching, mismatching, and gap penalties will be the SP score. And in multiple sequence alignment, what the algorithm is trying to do is to get the alignment which has the maximum this SP score. If the SP score is high, that means the matching is high. So the algorithm is trying to generate a sequence alignment, multiple sequence alignment with the maximum SP score. There are different methods for alignment. One is a graphical approach, and it's algorithm based approach. In the graphical approach, the alignment is created by weighted graph, and the tracing is done to get the maximum weightage. There are these methods can be used for multiple sequence alignment in a very limited way, and there are algorithms which can use the graphical approach to get the multiple sequence alignment. But the thing is that it is very tedious process, and it need huge amount of processing time and speed to do this graphical approach in a large scale data. And most easily done alignment method is algorithm based. It is trying to get maximum score and correctness of the alignment. And most of this algorithm is actually trying to do a replication of the evolution what could have been happened between two different sequences. We know that the frequency of substitution is high, and insertion of deletions are rare events, and there is a possibility of rearrangements. So all these things are taken into consideration when this algorithm is used for these algorithms are used for finding the matching between two different sequences. So it is not just matching between sequences, it is actually considering the evolutionary process and the evolutionary possibilities and the probabilities. So first method of this alignment, multiple sequence alignment, is exhaustive algorithm. As we know that it actually examine all possible alignment possibilities at the same time. So it use dynamic programming for aligning different sequences. It's a multi-dimensional search matching. So to align n sequences, they use n-dimensional matrix. As the number of sequences increases, it will become very difficult to handle that much amount of data. So, but it will provide the best result and best possible matches will be found by this exhaustive algorithm. DZA is a method under exhaustive algorithm. Next one is heuristic algorithm. Unlike the exhaustive algorithm, it do not guarantee the best result. But the thing is that it will provide the approximation and it results optimal solution will be obtained. Uh, but not the accurate algorithm. But when we are searching within or trying to match thousands of sequences together, it's better to use heuristic algorithm rather than exhaustive algorithm, because in exhaustive algorithm it is actually considering all possibilities and it will take long time and a huge amount of processing power. But in heuristic algorithm, this method is way much simpler than the exhaustive method, and it will provide optimal solutions in minimum time. And there are different methods coming under heuristic algorithm, which include progressive alignment type, iterative alignment type, and block-based alignment type. I will give a brief introduction about all this method. In progressive alignment type, depending on step-by assembly of multiple sequence alignment, it is speed up the alignment in a multi-step process. So what it do, in the figure you can see, it has multiple sequences. They are comparing them, doing a pairwise alignment using the Needleman-Wunsch method, and after that they will create a tree. This tree is called a guide tree. So this first method, they will get an approximate idea of which sequence is more related to the other sequences. So based on that, they will again, based on the guide tree, they will again do a Needleman-Wunsch algorithm based sequence pairwise alignment, and they will refine the process. So this is a multi-step process. First they align and make the gate tree, and based on the gate tree they will again align, and they are trying to produce to create the best result. Example of this progressive alignment method is tea coffee, both tea and coffee, because they are doing global and local alignment at the same time. That's why it's called the tea coffee method. They use the global alignment with the cluster and local alignment with the legging. Uh, Local global alignments are pooled to calculate the consistency scores, and distance matrix and guide trees is based on the refined pairwise alignment. Next method is iterative alignment type. It is based on the idea that optimal solution can be found by repeatedly modifying the existing suboptimal solutions. So we will get a average solution and we will do it again and again to modify it and get the best solution. So this is the graphical representation. These are the sequence to be compared. First they will do a pairwise alignment to make a guide tree, and based on the guide tree they will give different weightages for the sequences and realign them using the dynamic programming, and this process is repeated again and again to get the best tree. And as we are doing it more and more time, the tree will get more and more refined. So we can actually stop whenever we wanted, but if we do it again and again, or more and more time is spent on this, the tree will become more and more accurate. That's why it's called uh, repeated process. Repeated process will actually increase the accuracy of the tree. And finally, we have the block-based alignment type. It is one of the most widely used method. We have seen this. This is a type of word method or something like that. We are converting the query into small words, and we are actually searching for these words. The Google search is using the same strategy, and most of the database searches are using the same strategy to get the solution in the minimum time. So each genome is divided into distinct blocks with equal size, maybe 3 nucleotide length or 3 amino acid length, and these blocks are compared with the algorithm or the database. Whole database, the blocks are compared. Once they found a uncapped alignment among these blocks, they will find that and they will refine the sequence alignment. So that's how the block-based alignment work, and the matchbox is an example of block-based alignment algorithm. Thank you.